Welcome to myacademy.com. I'm Professor Mahfooz from Pakistan. This talks number 13 is about the classification of bacteria and fungi and the gram staining. Possible questions are, what is gram staining? So you have to describe that. What is it used for? Steps of the gram staining, classification of the bacteria, and classification of the fungi. So let's start. Now, how do we do gram staining? Remember the mnemonic? Come in and stain. We use the crystal violet stain first, then the iodine treatment, then the alcohol treatment, and then stain with the safranin. So all the bacteria are here. The first of all, the crystal violet, so they all turn into violet color. Then the iodine treatment, then the decolorization with the alcohol, and then the counter stain with the safranin. If the bacteria takes the safranin stain, they are called gram negative, and if they retain the original crystal violet stain, they are called gram positive. So that's how we describe or divide bacteria into gram positive and gram negative. First of all, classification of bacteria. Bacteria are classified into 5 main groups according to their basic shape. If they are spherical or round, they are called cocci. If they are like rods or flat, they are called bacilli. If they are spiral, they are called spirilla. If they are comma-shaped, they are called vibrios. And if they are corkscrew type, they are called spirochetes. Now, we will be mainly dealing with the cocci and the bacilli. So what you need to concentrate is that if the cocci are gram positive, they are called gram positive cocci, and if they are gram negative, they are called gram negative cocci. We'll discuss that in more detail in coming slides. The rods can be gram positive or gram negative depending whether they can take the gram stain or not. The prime examples of gram-positive cocci are Staphylococcus aureus, Staphylococcus epidermidis, and Staphylococcus pneumoniae. And the gram-positive rods are diphtheria and Corynebacterium cirrhosis, while the gram-negative rods are Pseudomonas aeruginosa and Enterobacter, which are Klebsiella, Serrata, Proteus, and E. coli. So these are the main bacteria which are involved in ophthalmic infections, and we'll describe now how to differentiate into gram-negative and gram-positive. Classification of the fungus is a bit confusing, and there are many terms used like yeast, mold, unicellular, multicellular, dimorphic, filamentous, hyphae, branches, mycelium, and spores. I've tried to make it relatively simple for you. Fungi, which are unicellular, are called yeast, and fungi which are multicellular are called molds. Now, next I will tell you the different forms of mold or different terminology used for mold. The mold is usually a branching structure, and the branches keep branching into smaller and smaller branches. So you can call these branches filaments, you can call them hyphae, you can call them branches, or you can call them mycelium. So all these 4 or 5 terms are used for the molds. So the molds can be filamentous or branches or mycelium or hyphae. You can use any term, and different books use different terms. Then some of the fungi are dimorphic, which means under certain conditions they are unicellular, and under certain conditions they are multicellulars. So both yeast and mold form spores during the asexual production, so the spores can be formed by the unicellular or multicellular. I hope that has made classification a bit easier. For an ophthalmologist, what you need to remember that fungi can be filamentous or non-filamentous. Filamentous can be septate or non-septate, and the septate then can be non-pigmented or pigmented. So first of all, first group is filamentous, septate, non-pigmented, also called hyaline or translucent, and the main examples are Fusarium and Aspergillus. These two are responsible for almost 98% of the fungal infections in the eye, so you need to remember Fusarium, Aspergillus, which are filamentous, septate and non-pigmented. The second group is filamentous, septate and pigmented, also called Demeticus or Phaeoid. Examples are Alternaria and Curvularia. The third group is filamentous non-septate, means they don't have any septa in their filaments or branches. The main example is mucor, and it causes mucormycosis. The fourth group is yeast, and the prime example is Candida. Thank you very much for being with me, and do visit youtube.com at Professor Mahfooz 7544, and please do give your feedback. Thank you.